Hi, this is Les and Amy from the RevGenetics Podcast. You can listen to the podcast on YouTube and now on Spotify, where we do a deep dive on supplements and longevity studies.
Yeah, and today we are getting into something really wild.
We really are. We're looking at a recent study that shows how a simple visual trait, in fruit flies specifically, the changing color of their eyes, acts as a literal aging clock.
Right, it's an actual visual clock.
It's crazy. It not only shows how old their cells are on the inside, but it can actually predict how long that specific fly is going to live.
I mean, it is just a completely fascinating piece of research. Because what we are really talking about here is finding a window into the microscopic decay of a living cell.
Yeah.
Like, we have always known that aging happens at the level of our DNA, right? But being able to watch it happen in real time, just with the naked eye, it completely opens up how we understand cell degradation.
And more importantly, how we might actually slow that degradation down. So let's unpack this for you, because the core mystery here for you, the listener, is this. What if you could look at a simple mosaic pattern on an animal and just instantly know how fast its cells are aging?
Mm-hmm.
And what if tweaking its diet or just the temperature of the room it's in could visibly rewind that clock right before your eyes?
It sounds like science fiction, honestly.
It really does. But to get there, we have to understand the underlying mechanism. We need to talk about heterochromatin, which is just a fancy science term for tightly packed DNA.
Right. So to really understand heterochromatin, just picture the inside of a cell. Your DNA isn't just, floating around loosely in there like a bowl of spaghetti.
Right. It's not just a big mess.
No, it is highly organized. A huge portion of it is spooled up and bound super tightly. And this densely packed DNA is the heterochromatin.
And the really important job of those tight bundles is silencing, right?
Exactly. Whatever genes are wrapped up in those bundles, the cell just cannot read them. They're effectively turned completely off.
Okay, so think of your DNA like a massive physical library.
Yeah.
You've got millions of instruction manuals in there. That tightly packed heterochromatin is basically these sealed, locked-away archive boxes deep down in the basement.
That's a great way to picture it.
And it's not just storing, useful things that you don't need right now. A lot of what is in those locked boxes is chaotic or even dangerous.
Oh, absolutely.
We're talking about vast stretches of junk DNA and these ancient virus-like genetic codes that have just embedded themselves in us over millions of years.
Right, the retrotransposons.
Exactly. You absolutely do not want your cells reading those particular manuals.
You really don't. And a great example in this study is an ancient virus-like element in the fruit fly called copia.
Copia?
Yeah. Normally, it's locked securely in that basement. But if it gets out, it acts like a rogue copy and paste machine. It just starts inserting itself randomly everywhere, triggering massive inflammation.
Yikes. So if I'm understanding this right, does aging basically just mean that those archive boxes in the basement are breaking open?
Yes, that is precisely it. That's called the heterochromatin loss model of aging.
Right.
As you get older, the biological tape holding those archive boxes together just starts to degrade. The DNA physically loosens up.
And when that happens, the bad stuff wakes up.
Exactly. The silenced genes, including those destructive virus-like ones, they wake up, flood the cell with chaotic signals, and that cellular chaos eventually leads to tissue breakdown and, mortality.
Wow. But here is the massive hurdle the researchers faced.
Yeah.
You can't just look at a living creature and watch its microscopic DNA unraveling.
No, you can't see the archive boxes breaking open.
So to visualize this invisible decay, they used regular lab fruit flies. And they used this genetic trick called position effect variegation, or PEV.
Right, which is just a brilliant methodology. PEV is basically a genetic trick that lets scientists literally see the boundary lines of that locked up DNA.
How did that work?
Well, they look at a specific gene in the fly called the white gene. Now, ironically, in a normal fruit fly, the white gene actually produces red pigment for their eyes.
Okay, wait, so the white gene makes the eyes red.
Yeah, genetics can be very confusing with naming. If the gene is broken, the eyes are white, so they called it the white gene decades ago. But normally, it makes red eyes.
Got it. So we have a gene that makes eyes red, and the researchers physically moved this gene, right?
Exactly. They parked it dangerously close to those tightly taped archive boxes.
Right on the boundary line.
Yeah. And because it's parked so close, the red eye gene accidentally gets caught up in the packaging. It gets dragged down into the basement and silenced.
But the boundary isn't a perfectly straight wall, is it?
No, it shifts slightly from cell to cell. So in some eye cells, the gene gets swallowed up and silenced, creating a white patch. In other cells, it stays free and makes red pigment.
resulting in a fly born with mosaic speckled eyes, just patches of red mixed with patches of white.
Exactly.
So the white patches are the physical spots where the red eye gene successfully got locked up.
Yes.
And here's where we get to the actual clock part of this deep dive.
Yeah.
They didn't just look at these speckled flies once. They tracked the exact same flies as they got older over 4 weeks. And what they saw was wild. As the flies aged, the white patches in their eyes slowly disappeared. The eyes just got progressively more and more red.
And what's really important is that they proved this wasn't just like leftover pigment building up over time.
Right.
They ran specialized lab tests and proved the DNA was actually unzipping. The cellular machinery was actively reading genes that used to be locked away.
So when you look at those eyes turning red, you are witnessing the literal, real-time unraveling of the DNA packaging.
You are.
That is incredible. But, this feels like a perfect moment to take a quick pause.
It is. And naturally, all this uncovering of longevity science is made possible by RevGenetics.
They really do have an incredible long-term focus on evidence-based natural compounds for aging research, which is exactly the kind of cellular support we're talking about today.
Oh, absolutely. Taking a science-first approach with well-researched compounds like resveratrol and pure NMN helps support that fundamental cellular health we all want as we age.
Exactly. Okay, so getting back to the deep dive. The researchers realized that if this eye color thing is a clock, maybe they could use it to tell the future.
Right. They wanted to predict lifespan.
Because even at just 2 days old, not all flies in a population look identical. Some are born with much whiter eyes, meaning they have super tight, secure DNA packing right from the start.
And others are born with redder eyes, meaning their biological tape is already a bit loose.
So they separated these 2-day-old flies based purely on their eye color. The white-eyed group with tight packing, and the red-eyed group with loose packing.
And the results were just staggering.
Yeah.
The group with the tightly packed white eyes lived a median of about 17 days.
Right.
But the looser red-eyed group lived only about 13 days. That is roughly a 30% increase in lifespan.
30%. That is massive. And they predicted it entirely by just looking at their eyes on day 2.
I mean, translating that to human terms, that's the difference between living to 70 versus making it well into your 90s.
Exactly. And they didn't just live longer, they lived healthier. The researchers gave them basically a fly fitness test, seeing how fast they could climb up a tube.
Right, negative geotaxis.
Yeah, and the white-eyed flies with the tight DNA packing stayed much faster and stronger as they got older.
Because their cellular basements were perfectly organized, their nervous systems actually stayed physically younger.
So tight boxes equal a longer, healthier life. And this is what you, the listener, are definitely wondering right now. Can we control this?
We can try.
Like, how did the researchers try to actively hack this biological clock?
Well, they tried 2 main environments. First, they gave the flies a low-nutrient diet, basically fasting them by cutting their food in half.
Oh, wow. And the second was temperature, right?
Yeah, they kept another group at 18 degrees Celsius, which is about 64 degrees Fahrenheit, instead of their normal warm room.
So we have fasting flies and we have cold flies.
Right. And in both cases, the stress triggered a massive biological response. The fasting flies and the cold flies both developed significantly whiter eyes.
And they lived longer.
They did.
So just by giving them less food or making them chilly, the cells realized, hey, resources are scarce, we need to conserve energy. So they double-taped the archive boxes shut.
Exactly. It's a protective survival state. And they actually proved they could trigger this exact same protective state genetically, too, using a mutation called the Chico gene.
Ah, the Chico gene.
Yeah.
That one comes up a lot in longevity research.
It does. It dials down the insulin signaling pathway. So it essentially tricks the fly's body into thinking it is fasting even when it's eating totally normally.
And their eyes stayed whiter and they lived longer, just like the flies they were actually fasting?
Precisely.
But wait. I'm looking at the next part of the study with the packing cape itself.
Mm-hmm.
And this is genuinely confusing.
You mean the HP1 mutation?
Yeah. So they mutate the gene that physically manufactures the packing tape for the DNA. They literally break the tape dispenser. Yeah. And as expected, the boxes burst open, the eyes turn dark red, the clock spins forward. But the flies didn't die faster.
No, they didn't.
If losing the tight packing causes aging and deaths, shouldn't those flies have aged super rapidly and died right away?
It is a brilliant contradiction. At first glance, it seems to completely break the model. But it actually reveals this profound hierarchy in how our biology works.
A hierarchy, like a chain of command.
Exactly. Think of things like the insulin pathway or your response to diet as the master commanders. They sit at the very top.
Okay.
When you fast, that master commander orchestrates a massive survival program. It triggers a longer life, and it orders the DNA to be tightly packed as a protective side effect.
Oh, I see. So the tight packing is just a downstream effect.
Yes. So when they manually broke the tape dispenser, they only broke a symptom of youth. They broke the clock on the wall, but they didn't break the master commander holding the actual power.
That makes so much sense. Smashing the thermometer doesn't change the temperature of the room.
Exactly.
So bringing this all home, why should you, listening to this, care about the eye color of a tiny fruit fly?
Because the exact same mechanisms of DNA unzipping and waking up dormant junk DNA and cellular inflammation that is happening inside you right now as you age.
And the really exciting part is that those master commander pathways, the ones that respond to stress, we have those too.
We do. Things like intermittent fasting or dietary restriction or even taking a cold plunge.
Yeah.
These are actively being studied in humans for these exact epigenetic benefits.
Right. When you do a cold plunge, you are trying to send that exact same survival signal to your cells. You're telling them to double tape your own genetic archive boxes.
And this study is one of the clearest visual proofs that those kinds of interventions actually work at the fundamental level of DNA.
Which leaves us with a truly mind-blowing thought for the future.
Oh, absolutely.
If a simple mosaic eye color can perfectly report the invisible decay of DNA inside a fly, what is the future of human biomarkers?
Right now, you have to get your blood drawn and sequence your DNA in an expensive lab to find out your biological age.
But what if we could engineer harmless, visible reporters in humans?
That would change everything.
Just imagine a tiny, engineered patch of skin on your arm, like a little freckle that subtly changes its pigment based on how fast your cells are aging.
You wouldn't have to guess if your new fasting routine was working. You could literally just look at your arm.
You could just watch your biological clock rewinding in real time. It's wild to think about.
It really is.
We hope you found our discussion of the published study interesting.
Thank you for listening to the RevGenetics Podcast.
RevGenetics.com for the best longevity supplements on Earth.